Hello, my name is Katie Gore and this is my final project on Alzheimer's disease. This is a video detailing different causations of Alzheimer's disease, as well as explaining the difference between both early onset and late onset Alzheimer's, further clarifying the reasons some patients develop Alzheimer's disease earlier in life than others. First, let's discuss the most common cause of Alzheimer's, the factor associated with nearly every patient of dementia: increasing age. Increasing age causes neurological problems in the brain related to Alzheimer's disease, such as increasing atrophy, inflammation, production of unstable molecules called free radicals, and breakdown of energy production within the cells, all in the brain. These problems developing in the brain are likely related to mutations in the genes inherited from chromosomes passed down from parents. If a person has a parent that has dementia in some form, the person has a higher risk of developing the disease as well. Genetic variants of genes can also occur that cause the disease directly or create issues that can indirectly lead to Alzheimer's disease. Now let's discuss the differences between late onset and early onset Alzheimer's disease. Early onset can appear between the ages of 30 and 60, is very rare, and is related to gene changes passed down from parent to child. Late onset appears typically age 65 and above, is the most common type, and may involve a gene called apolipoprotein E4. Late onset does not have a definitive cause, but the appearance of the gene apolipoprotein E4 on chromosome 19 does seem to increase the risk. Inheriting the gene allele does not guarantee Alzheimer's disease, however. Interestingly, apolipoprotein E2 seems to decrease the risk of Alzheimer's, while apolipoprotein E3 has a neutral effect on the disease. Some early onset cases are caused by an inherited change in one of 3 genes, resulting in a type of early onset familial Alzheimer's disease, or FAD. A child whose mother or father carries a genetic mutation for early onset FAD has a 50-50 chance of inheriting that mutation. If the mutation is in fact inherited, the child has a very high risk of developing early onset FAD. A study conducted by a group of psychologists in London studied the prevalence of early onset Alzheimer's disease and found that 54 out of 100,000 people ages 35 to 65 developed Alzheimer's disease, and that every 5 years that amount doubled. Early onset FAD is caused by any one of a number of different single gene mutations on chromosomes 21, 14, and 1. Each of these mutations causes abnormal proteins to be formed. Mutations on chromosome 21 cause the formation of abnormal amyloid precursor protein, APP. A mutation on chromosome 14 causes abnormal presenilin 1 to be made, and a mutation on chromosome 1 leads to abnormal presenilin 2. A study done in 1992 by John Hardy and Gerald Higgins hypothesized that the amyloid plaques are what cause the other features of Alzheimer's disease, such as neurofibrillary tangles, vascular damage from extensive plaque deposition, and neuronal cell loss. These amyloid plaques are caused by the deposition of A-beta-P. The breakdown of the APP protein is related to the deposition of A-beta-P, and that mutations in the COOH terminal portion of APP causes hereditary early-onset Alzheimer's disease. A-beta-P is an amyloid protein that forms extracellular insoluble deposits and is attached to APP at the terminal sequence. APP is cleaved typically at the A-beta-P sequence, producing fragments that don't contain A-beta-P and can't cause deposition. When this cleavage is mutated and doesn't occur normally, A-beta-P can build up and cause amyloid plaques. These plaques are associated with Alzheimer's disease. There is evidence that either A-beta-P containing COOH terminal fragments is toxic to cultured neurons, or it renders neurons more sensitive to excitotoxic damage. The peptide is known to disrupt calcium homeostasis and increase intraneuronal calcium concentrations. This observation could explain how neurofibrillary tangles form. The tangles are largely composed of paired helical filaments formed from a hyperphosphorylated form of the microtubule-associated protein tau. And tau phosphorylation can be controlled by intracellular calcium. There are many different causes of Alzheimer's disease, but these are just a few reasons Alzheimer's disease occurs. Thank you for watching.